So, looks like a lot. We'll break it down step by step. Just as a start, all living cells have membrane potentials, or electrical gradients, that are across their membranes. Okay? Inside the membrane is usually around -60 to -90 millivolts relative to the outside. This is referred to as a cell's membrane potential. Neurons and muscle cells can use their membrane potentials to generate electrical signals. This is done by moving ions across their membranes to create electrical currents. The ions move across membranes in ion channels. In this scenario, we are looking at acetylcholine. See this? Synaptic vesicle containing acetylcholine. So this neuron, see, you can see the myelin sheath around the axon, attaches to the skeletal muscle. We're zooming in right here. Break it down. We got a synaptic vesicle. It's got acetylcholine, a chemical in there, an ion. So this acetylcholine gets shot out, exported outside of the neuron into this neuromuscular junction. Okay? And oops, I'm sorry. I didn't mean to click. So the acetylcholine is in the neuromuscular junction. The acetylcholine can go on to, the arrow goes down and up here. Acetylcholine can go down and attach onto the protein, so the receptor. So the acetylcholine is the ligand in this scenario. It attaches to the receptor, opens up the protein, and sodium that was present in this channel can now flood through. The positively charged ion of sodium changes the inside potential of this muscle cell to be more positive. All right? So it's going from -90 millivolts to -60. So it's changing the electrical gradient of this muscle fiber by pushing acetylcholine into, or I guess exporting acetylcholine into the neuromuscular junction. It attaches to the receptors and allows sodium to go through, which changes the charge inside the membrane here. So what we're looking at is called a neuromuscular junction. It's where the terminal end of the neuron meets the muscle fiber, and thus is a junction of neuron and muscle. The coupling of the two properties of excitation and contraction is the basis of how muscles move. There is excitation of the muscle fiber by the motor neuron that elicits a contraction of the muscle fiber. So at this point, we have accomplished step 1 of our previous slide. I'm going to go back really quick. We have activated our skeletal muscle by the nerve. Okay? Activation of the muscle cell has been accomplished by the nerve. Step 1 achieved. So excitation-contraction coupling seems like a daunting term at first. Your textbook says it, I'll say it, seems like a lot. But if we break it down, it simply means in order for the muscle fiber to contract, its membrane must be first excited. In other words, it must be stimulated to fire an action potential. The stimulation here was that sodium that passed through sodium channels into the cell. This increased the cell's membrane potential, and that elicited an electrical stimulation or impulse. See, it says right here, the release of acetylcholine at the neuromuscular junction caused an electrical impulse to be generated in the muscle cell plasma membrane. So acetylcholine was released in there that let sodium go through, and that changed the membrane potential, sending an action potential throughout the sarcolemma of the muscle cell. The electrical impulse then is carried through the cell's interior by T tubules, transverse tubules. That's what T means, is transverse. So this would be along the cell, and this would be transverse to the cell. These T tubules, these lines that go down, carry the electrical impulse, and this electrical stimulation then triggers calcium to be released from the sarcoplasmic reticulum. This blue lining the outside of the particular myofibrils, that's where the calcium is stored, remember? And so this electrical impulse then elicits the calcium to be released from the sarcoplasmic reticulum. And so at this point, we have accomplished step 2 of our basic steps of muscle contraction. So let's go back here real quick. Step 2, nerve activation increases the concentration of calcium ions. Sorry, there's a dumpster going off behind me. Nerve activation increases the concentration of calcium ions in the vicinity of the contractile proteins. Okay, so calcium ions have thus been released. Okay? So, Activation releases calcium. This is contraction. The formation of cross bridges between thin and thick filaments, the actin and myosin. So just some quick definitions here. Tropomyosin is it covers the binding sites on actin. It's a protein that covers the binding sites on actin. And it's basically saying that myosin cannot bind to actin. Troponin is another protein, and it has a binding site for calcium. Troponin and tropomyosin are kind of complex together. They regulate whether or not actin and myosin can interact. When actin and myosin do interact, they contract, thus letting the muscle move. They, in a normal resting state, do not allow that. And tropomyosin, troponin does not allow the actin-myosin interaction in a resting state because calcium has to be present for cross bridges to form. And the cross bridges, again, are actin and myosin reacting with each other. In the absence of calcium, or a resting state, non-contracting state, the protein complex of troponin, tropomyosin, covers the myosin binding sites that are on actin. So the troponin, tropomyosin covers the site on myosin. It covers the site on actin where myosin could bind. So basically, troponin and tropomyosin does not allow myosin and actin to interact with each other if there's not calcium. But if we have calcium, bam, troponin and tropomyosin react differently, and myosin and actin can interact. And so that is shown here. What we're looking at are the thin and thick filaments and how they interact. Again, this all looks incredibly daunting. I understand that. You have to take it step by step, listen to how I'm presenting it, and then go back, diagram things out, write the steps. It will make sense. It's just a lot to absorb with just one lecture. You have to go back and look at this stuff though. So again, calcium is initiating the sliding filament reaction. All that means is we're allowing actin and myosin to react with each other. So this is our relaxed or resting state. This is our contracting state. In the relaxed and resting state, we do not see actin and myosin touching each other. They're not touching each other at all. That myosin heads do not make contact with actin. But once calcium is released from the sarcoplasmic reticulum, calcium then binds to troponin. Troponin, tropomyosin complex, shifts its position, and it allows the myosin binding site on actin to be revealed. Okay? The shift in the position exposes the binding site in the myosin molecule. Pulls the actin filaments toward the center of the sarcomere. Okay? So, the myosin heads form cross bridges with the actin, and those are simply when they make contact. That's all the cross bridges means. Actin filaments are pulled toward the center of a sarcomere. Sarcomere shortens. All the sarcomeres shortening lead to the entire muscle contracting. So they shorten, contraction. So think of your bicep coming. You have your arm extended, and then you contract and flex your bicep. This is what's happening with all these myofibrils. They're all shortening, bringing your wrist and your outside part of your arm closer to your shoulder. And so here are the steps that are laid out kind of on the previous slide. We have calcium initiating the sliding filament mechanism. Calcium is released from the sarcoplasmic reticulum. Calcium binds to troponin. Troponin-tropomyosin complex shifts its position, revealing the myosin binding site. Myosin heads then form cross bridges with actin. Actin filaments are pulled toward the center of the sarcomere.